So thank you very much for this kind invitation. It is an honor, and I'm deeply touched to participate to the John Gerber Memorial Lecture. I would like to thank the organizing committee for inviting me. So the work in our laboratory over the past many years has primarily focused in investigating mechanisms of antiretroviral drug transport across blood tissue barriers, as well as at HIV sites recognized as sanctuary and cellular reservoirs. Today, I would like to share some key findings from our work. So pharmacological treatment of HIV, as we all know, is very complex. It consists in the combination of several drugs belonging to different classes of pharmacological agents that target different stages of the HIV cycle. So the therapy, as we know, is extremely effective in almost eradicating the virus from the periphery. However, it does not offer a cure and presents a number of limitations. And this is due in part to the fact that most of these drugs are extensively metabolized, as well as they can undergo transport across cell membranes and tissue sites that can result in a number of drug-drug interactions and adverse effects. So if we further address drug transporters and drug metabolic enzymes, as we know, they are extremely important in contributing to the overall drug disposition in mammalian systems. In particular, drug transporters belonging to the solid carrier transporter superfamily are extremely important in facilitating the intake or uptake of various xenobiotics, including drugs and antiretroviral drugs, into target cells and tissues. Members of the ATP binding cassette membrane transporter superfamily, on the other end, are going to actively pump out compounds that were able to cross the lipid bilayer using direct ATP energy. So this is a very effective mechanism of cell protection against a number of xenobiotics. And this effect is also known and responsible for multi-drug resistance, particularly in cancer chemotherapy and also in antiviral therapy. Members of the ATP binding cassette membrane transporters include P-glycoprotein, breast cancer resistant protein, and multi-drug resistant proteins. Anti-HIV drugs can be very good substrates for all of these efflux transporters, but also at the same time, they can inhibit some of these transporters and also exert induction. And the induction is due to the fact that a number of antiretroviral drugs are also very effective ligands of a number of transcription factors, particularly nuclear receptors, that once activated by the ligands, then will increase the transcription of the same transport protein. So we can see a fair amount of complexity and in the interplay of the effect of these transport proteins in regulating cellular concentrations. In addition, when compounds reach the cytosol, then they can be further degraded, so metabolized by a number of metabolic enzymes, particularly those belonging to the cytochrome P450. So drug metabolism is well known to contribute to overall drug clearance and also overall drug disposition. It is well known that a number of anti-HIV drugs, particularly HIV protease inhibitors, are also good substrates, but also inhibitors of a number of cytochrome P450s. So this will need as well to be taken into account in terms of tissue concentrations that will reach target sites. So as we were mentioning, although the pharmacology of HIV is very effective, in controlling the infection, it does not cure it. And the reason for no cure to HIV is the fact that the virus can persist and hide in a number of tissue sanctuaries, such as the brain and the testes, both protected by blood tissue barriers, and we will come back to that, as well as to the fact that there are a number of viral reservoirs that have been identified, particularly the resting T lymphocytes, the memory cell subsets, gut-associated lymphatics, and renal epithelial cells. So our laboratory has primarily focused in understanding mechanisms of permeability of anti-HIV drugs across blood tissue barriers. And I will discuss today primarily the blood-brain barrier and the blood-testis barrier. We've looked also at the blood-intestinal barrier and more recently at the placental barrier. So the blood-brain barrier, as you know, is primarily formed of endothelial, microvessel endothelial cells. Morphologically, they present very tight junctions between the cells that prevent significant paracellular pathways of trafficking between the periphery and the brain parenchyma. So this is the hallmark of the morphology of this vasculature. Therefore, at the level of the membranes of the endothelial cells, we see very high expression of membrane transporters that are going to permit the trafficking of nutrients, endogenous compounds such as hormones, as well as xenobiotic across the barrier between the periphery and brain parenchyma. It's important to indicate that the close proximity of pericytes, astrocyte food processes, microglia, and axonal processes to the endothelial cells is very important in maintaining the integrity of the barrier. Also to indicate, as we are going to discuss, that in terms of the transporters, we are going to see very high expression of P-glycoprotein and breast cancer resistant protein at the level of these cells, forming a biochemical barrier that is very effective in preventing the permeability of a number of anti-HIV drugs. The blood-testis barrier presents a lot of similarities to the blood-brain barrier and is one of the tightest barrier in mammalian systems. It is formed by epithelial Sertoli cells that, like the brain microvessel endothelial cells, show very tight junctions between the cells that, again, present a physical barrier. At the same time, it is, from our work, we've demonstrated that we have high expression of membrane efflux transporters forming a biochemical effective barrier. These Sertoli cells line the lumen of the seminiferous tubules, then offer protection for the spermatogenesis function. So we've been interested in examining permeability of anti-HIV drugs at these tissue barriers. So clinically, it's interesting to put together the data from antiretroviral drug concentrations reported for a number of different classes of anti-HIV drugs. And the CSF to plasma concentration ratio, as well as semen to plasma concentration reported in the clinic, shows that for protease inhibitors, these ratios are very, very low. And this is very interesting and reflecting the fact that HIV protease inhibitors are known to be very good substrates for efflux transporters. So here are some key findings from our work. This is now almost 20 years ago when we reported pretty much for the first time the localization. We also did expression and function of P-glycoprotein at the rodent blood-brain barrier and also in human blood-brain barrier. So we see here data at the electron microscope of immunogold cytochemistry using specific antibodies to P-glycoprotein. So the gold particles show the localization of P-glycoprotein. And we demonstrated that the transporter transporter is localized at the level of the luminal membrane, at the abluminal side in pericytes, as well as the astrocyte. So all along of the barrier, which is called the neurovascular unit. In primary cultures of astrocytes isolated from human and rodent brain, we also demonstrated localization of P-glycoprotein at the cell surface, at the level of the astrocyte processes, and also at the cell surface in caveoli. We also showed a subcellular localization at the nuclear envelope. In microglia cells, which are the primary immune cells of the brain and the primary target of HIV infection in the brain, we also demonstrated localization of P-glycoprotein at the plasma membrane and also subcellularly. These were very important, very novel results demonstrating that the transporter is localized in the target cells of the infection in the brain. So in summary, I cannot present all of the data. P-glycoprotein functions as a very effective efflux mechanism that will restrict brain entry of several xenobiotics, including anti-HIV drugs. The transporter is not only expressed at the level of the vasculature, but also at the level of the brain parenchyma. Therefore, here forming a secondary biochemical barrier to the primary barrier at the level of the vasculature. Moving on to testicular tissue studies, Kevin Robillard, a doctoral student in my laboratory, initiated the work looking at human and rodent Sertoli cell culture systems and working with transgenic in vivo mouse models. So we work with Sertoli cell culture models, mouse model, a cell line, the TM4, and we also work with human primary cell culture Sertoli cells. In these cells, applying immunocytochemistry with immunofluorescence, we showed nice localization of P-glycoprotein, multi-drug resistant protein 1 and 4, and breast cancer resistant protein. We use the sodium potassium ATPase cell surface marker to show the localization at the level of the cell membrane. And here we have the merged images, and similar data was demonstrated as well in the human Sertoli cells. So these were exciting results. We followed these with detailed function analysis for the transporter protein and their role in preventing the transport of a number of HIV protease inhibitors. We moved on into in vivo studies, and these were performed in a triple knockout mouse model lacking P-glycoprotein and breast cancer resistant protein. And we performed a pharmacokinetic study using atazanavir. So what we showed here are the brain to plasma concentration ratio of atazanavir, as well as the testicular to plasma concentration ratio for the protease inhibitor. So these ratios, as we see depicted in red in the triple knockout mouse model, are significantly higher in the brain and in the testis. This directly demonstrating the in vivo role of the transporters in restricting the distribution and permeability of the drug into the tissues. But this was all good and done in vitro and in vivo, so very preclinical, and we needed to see more translational data. So in collaboration with Jean-Pierre Routy at the McGill Health Center in Montreal, and we were very fortunate to have his involvement, he was able to provide us human testicular tissue from HIV-infected donors and non-infected controls. This is a very rare tissue to obtain, and we obtained the tissue from individuals undergoing orchiectomy for gender reassignment. Billy Wang and Sanake White-Allman undertook this work in my lab. So in the testicular tissue of the donors, we see localization of P-glycoprotein, multi-drug resistant protein, and breast cancer resistant protein at the level of the seminiferous tubule. So this was very exciting to observe. We also documented the localization of a number of SLC transporters and also a number of cytochrome P450 enzymes. In collaboration with Dr. Courtney Fletcher at the University of Nebraska, we were able to quantify antiretroviral concentrations in these testicular tissues from the HIV-infected donors. And we observed that for the HIV protease inhibitor darunavir, we had testis to plasma concentration ratios that were extremely low at 0.2. So very low testicular concentration, suboptimal. We also show similar low concentrations for efavirenz at a ratio of 0.2. So darunavir is a high affinity substrate for P-glycoprotein. Efavirenz is a good substrate for breast cancer resistant protein. So together, these data suggest the role of these transporters in the human testicular tissue. So in conclusion, from the in vitro and in vivo studies, we've demonstrated expression and activity of drug efflux transporters in models of the blood-brain barrier, brain parenchyma, and also at the level of the blood-testis barrier. In vivo studies in the transgenic mouse model demonstrated the direct effect of the drug efflux transporters, particularly PGP and breast cancer resistant protein, in restricting atazanavir distribution in the brain and testis. We also generated novel data on drug transporters and metabolic enzyme expression and localization in human testicular tissue. And more recently, we showed similar expression at the level of T lymphocytes isolated from the testicular tissues. Differences between plasma and testis antiretroviral drug concentration appear to reflect expression activity of these drug efflux transporters. Finally, we can conclude that the testes are a complex pharmacological compartment which can limit the penetration of certain antiretroviral drugs and that it could contribute to HIV persistence similarly to the brain. So I would like to acknowledge the contribution of many trainees in my laboratory over the past many years. I'm going to show various groups here without having time to identify them. This is the most recent group, actually just before COVID, and acknowledge the collaboration of several HIV researchers and their group, in particular, Yuri Perstitsky at Temple University, Rupert Cole in our own institution, Courtney Fletcher at the University of Nebraska, David Volsky at Columbia and Icahn School of Medicine, Jean-Pierre Routy at McGill University, and Petronella Kunta at the University of Montreal. I want to acknowledge the longstanding support from various funding agencies in Canada, Canadian Institute of Health Research, Canadian Foundation for HIV Research, CanCure, HIV Cure Program at CIHR, and the Ontario HIV Treatment Network. Thank you so much for providing me this opportunity to share some of this work. I'll be happy to share some questions.